In this video, we will create an animation of protein synthesis that explains how a person could end up with trimethylamminuria, or TMAU. TMAU is known to be caused by the C to T substitution mutation in the first letter of the fifth triplet on the antisense strand of the FMO3 gene.
Transcription occurs in the nucleus where a strand of DNA is transcribed into RNA. Transcription can be classified into three parts: initiation, elongation, and termination. RNA polymerase binds to the promoter region before the beginning of the gene on the DNA template strand. The other DNA strand that does not bind is called the sense strand, also known as the non-template strand or the coding strand. This step is called the initiation of transcription. RNA polymerase is the enzyme responsible for unzipping the DNA and transcribing new mRNA strands. The DNA is read from the 3' to the 5' end of the antisense strand. At the active site, RNA polymerase creates hydrogen bonds between the antisense strand DNA nucleotide and the RNA nucleotide. Also, RNA polymerase covalently binds the mRNA nucleotides together in a reaction that involves the release of the two additional phosphates. This is called the elongation. The RNA polymerase moves along the coding sequence, synthesizing RNA from the 5' to the 3' direction. As seen in the simulation, the mRNA nucleotides binds to the antisense or the template strand. The mRNAs that bind at the active site are complementary to the antisense strand.
The mRNA nucleotides are very similar to that of the DNA, but instead of thymine, there is uracil. Hence, the mRNA nucleotides are adenine, uracil, guanine, and cytosine.
After going through transcription, the antisense and the sense strands of the DNA form new hydrogen bonds after the RNA polymerase passes by. The process of transcription continues until the RNA polymerase gets to a sequence of DNA known as the termination sequence. The RNA polymerase, the mRNA strand, and the DNA strand separate from each other. This is called termination. The sense strands, or the coding strand, has a very similar sequence as the mRNA strand, besides the fact that the thymine in DNA is replaced with uracil in RNA. After transcription, the mRNA strand is modified to promote the stability of the RNA. The 5' cap is characterized by the modified guanine at the 5' end, and the poly A tail is characterized by 50 to 250 adenines at the 3' end. The mRNA also goes through mRNA splicing, where non-coding regions known as introns are edited out by spliceosomes, so that they are not translated into amino acids. The coding regions of the mRNA are known as exons, and they are transported into the cytoplasm.
Here is a comparison of the normal and the affected FMO3 DNA and the mRNA strands. Because of the C to T substitution mutation on the antisense strand, the resulting 13th nucleotide on the mRNA codon is adenine instead of guanine. After transcription, translation takes place in the cytoplasm, where ribosomes use mRNA transcripts to synthesize amino acids. The mRNA strand created during the process of transcription binds to a ribosome. The ribosome begins to slide over the mRNA until it reaches the start codon AUG, where a specific tRNA with a specific complementary anticodon can bind. The tRNA molecule with start anticodon UAC binds to the start codon on the mRNA AUG to have the first amino acid of the polypeptide, methionine, made at the P site of the ribosome. A second tRNA molecule with the appropriate anticodon binds to the second codon. The ribosome catalyzes the formation of a peptide bond between the two amino acids, creating a dipeptide carried by the second tRNA. The ribosome slides over the mRNA molecule from the 5' to 3' direction, leading to the release of the first tRNA and the binding of a new tRNA to the following codon. The growing polypeptide chain remains attached to the tRNA at the A site. Then, the tRNA in the P site moves to the E or exit site, where tRNA is released. The tRNA carrying the growing polypeptide chain now moves to the P site. The polypeptide chain keeps growing as this process is repeated until a stop codon is reached, at which point the ribosome comes off the mRNA. The polypeptide breaks away from the tRNA and can be folded and modified into a functional protein. Here is a version of the FMO3 polypeptide chain that has not been affected by TMAU. As you can see, due to the substitution of adenine instead of guanine, the amino acid lysine was produced instead of glutamic acid for the patient with TMAU. This is called a missense mutation due to the change in the amino acid sequence. tRNA is activated by aminoacyl tRNA transferase. This happens when tRNA and the corresponding amino acid attaches to the aminoacyl tRNA transferase enzyme. This process requires ATP for phosphorylation to take place. The enzyme binds to ATP, where ATP goes through phosphorylation and it releases PPI and turns into AMP. This releases energy, and the phosphorylation causes the tRNA and the corresponding amino acid to bind, creating aminoacyl tRNA, which is charged and ready for use.
Protein folding occurs in the endoplasmic reticulum, where proteins are folded into specific structures that determine their functions. The primary structure, or the first level of structure in proteins, is the sequence of amino acids in the order in which they are formed during protein synthesis. The order of the amino acids is referred to as the primary structure. The amino acid sequence can fold into a secondary structure of either an alpha helix or a beta pleated shape. The structures are folded through hydrogen bonds between the amino acids. The secondary structure can fold into a tertiary structure as well, which determines the overall 3D configuration of the protein. The shape is created by bonding between the R groups in order to give rise to the 3D shape. Quaternary structures is the association of two or more polypeptide chains to form a complex protein molecule. Only some proteins have this structure. Each polypeptide chain will have a certain primary structure, secondary structure, and possibly tertiary structure within a quaternary structure.